Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. Thanks for joining today's webinar. It's presented, like all of our webinar series, by ASCCT and also the European Society for Toxicology in Vitro. And I would encourage you to, if you'd like to learn more about either of our societies, head over to our websites so you can find out more about what we do and consider joining as a member of either one or both to help support some of the programs that we're engaged in. Both of our organizations host a number of activities to promote computational and in vitro toxicology. And you'll see in our introduction, we have a focus on training and other activities, and also on students and early career scientists. The webinar is going to be recorded, and the recording will be posted on the ASCCT website a few days from now, and likely a copy of the slides as well. The lines are muted. If you want to ask a question of the presenters, use the Q&A feature down in the bottom toolbar at the bottom of your screen. You just type a question in there, and then we'll address each presenter's questions after they speak. So we'll have a presenter and then a Q&A session, and then the second presenter in a Q&A session. If you have a technical difficulty, you can also reach us in the chat. I just wanted to mention first that we have some upcoming webinars. Our January webinar is already scheduled for the 21st, and we're going to be hearing a presentation from Stefan Fuller and Kirsten Reisinger about reconstructed skin, comet, and micronucleus assays. So it's very exciting. And then in February, we're going to have some more award winner webinars like the one that we're doing today. So now I'm going to turn it over to Helena Kandarova, President of ESTIV, for a couple of updates.
Thank you very much, Christy. Good morning or good afternoon, depending on from where you are connecting to this webinar. My pleasure to introduce you two big activities that we are planning to have in the year 2022. So the first one will be the ESTIV Applied Toxicology course. You know that it has been originally scheduled for 2021, but due to the pandemics, we decided to move it to June 6 to 11, 2022 to be able to organize it really in person. So it will be happening at the Luxembourg Institute of Science and Technology. The training course program is available on the website, and the registration module is just about to be finalized today, and tomorrow it will be available for the registration. So we warmly welcome you to register. It is mainly for the early career scientists, but also suitable for those who are switching from the in vivo field to the in vitro field, or for those who need to update their knowledge. Second page, please. The next activity, large one, is the ESTIV Congress that also has been rescheduled from 2020 to 2022, and the Congress will be happening on the same place as we originally planned in Barcelona. It will be in November 21 to 25, and the scientific program is already available on the webpage. Abstract submission and registration is open, and I welcome you to check what will be happening on the Congress. We have updated a little bit the program and also enlarged. We are scheduling 2 parallel sessions. So I hope that every one of you will find something interesting on the program, and I will be happy if you could consider participation at our activity. So now I give the word back to Christy.
Great, thanks, Helena. We're all really looking forward to that meeting finally happening. So today we have a treat for you. We have 2 presentations from what we call our award winners for 2021. Both ESTIV and ASCCT facilitate and award several best poster, best presentation, other kinds of awards throughout the year, either at our own meetings or at other meetings that take place to try to support early career scientists and students who are working in vitro and computational toxicology. Today, we are hearing from Dr. Agnes Karmaus. She is from Integrated Laboratory Systems and NICEATM, and she has won the Applied In Vitro Toxicology Award, which is this is the first time the ASCCT has given out this award, and we're grateful to AIVT for making it available at our annual meeting in October. It comes with a $300 award and a 1-year subscription to the journal, and it's meant to highlight applied work that stands to make a lasting impact on NAM's implementation. And the second award winner for today will be Saskia Galanhoek, and she is from IUF Dusseldorf. She won the Best Poster Award at the 2021 Eurotox Congress, awarded by ESTIV. It includes a prize of 500 euros and a free ESTIV membership and other promotional activities, and is awarded to recognize scientific and technical quality of the poster and the abstract and innovativeness in the area of in vitro and silico toxicology. And so today you're going to hear from both of these excellent presenters. First of all, Dr. Karmaus is a senior toxicologist, integrated laboratory systems. She received her PhD in biochemistry and toxicology from Michigan State University, and then did a postdoctoral fellowship at US EPA's National Center for Computational Toxicology, focusing on in vitro assay development for the ToxCast high-throughput screening program. Currently, her work focuses on the application of Tox21 and ToxCast data toward new approach methodologies in support of the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICEATM. And she's going to be presenting gaining context for high throughput in vitro data, annotation approaches, and visualization tools. I'm going to go ahead and introduce our second presenter now, and then we'll be able to flow through the presentations. So Saskia Galanhoek is currently a PhD candidate in Professor Ellen Frietz's lab at IUF Leibniz Research Institute for Environmental Medicine. She's going to be finishing in January of next year. She received both her bachelor's and master's in biology at the Heinrich Heine University, and she's currently continuing the work of her master's thesis as a doctoral student, which she's going to be presenting for us here today. And that work is on the human-induced pluripotent stem cell test as an alternative method for developmental toxicity testing. So with that, I will turn the floor over to Dr. Karmaus, and feel free to share your screen and go ahead.
Thank you. And thank you so much for this opportunity, and of course to IVT for the award. I'm excited to be able to share this work with another audience, so thank you to everyone who's joining today as well. Today I'm going to talk about how we can gain context for high throughput in vitro data, including annotation approaches and visualization tools that we've developed to hopefully help understand better how to use these data and what the data are. So as a brief background, there's a new challenge. As we develop new approach methodologies, we're pushing toxicology into a new big data world. It's not true big data, but for toxicologists, it really is. For a long time, the challenge was generating data, developing the in vitro assays that could give us screening level information that is toxicologically useful. Now we're generating millions of data points and concentration response for chemical testing. And we've jumped into a new challenge, which is how do I understand what all this data is, which data is useful for me, which data is good data, or for my applications is good data. And so this new challenge is what we're hoping to address with the work I'll be presenting today. So some of the questions, of course, are the quality, what can I use this data for? What are the limits of the data? And how do I understand what these assays are? And so to tackle addressing that challenge, there's kind of what I consider four different approaches that we've applied within our work. So the first is curating data. We want to make sure that we're able to help people search just what we consider to be really robust, instead of the user having to figure out, is this good, is this not good, how do I look at it? Every assay has different criteria. The second would be having experts review all these in vitro assays and conduct annotation efforts so that there can be more approachable terms, some what we call controlled vocabulary. So within a structured framework, some language that everybody can understand instead of very specialized or something that seems cryptic and only the developers can make sense of. The third would be to then layer this via tool. Can we provide this to users so that they can integrate multiple data, they can get curated data, they can see the annotations that make it approachable? What tools can we then plug this information into? And finally, offering some visualizations to allow for a quick summary view for you to be able to evaluate this in one glimpse without having to sort through really big spreadsheets and look at lots of numbers or something that might be more intensive. So today I'll cover these 4 points and how we went about it and what our tool and what our approach has been. Of course, there's lots of approaches. These data are available in multiple places. So I'll be talking today about the integrated chemical environment. We call it ICE. This is supporting the development of new methods for predicting toxicity by providing improved access to the data and offering tools that users can use to better conduct analyses with one click of a button without having to be experts or coding algorithms and programs to do complicated math. This is what the landing page looks like at the moment. There's always new updates and releases. There's lots of new tools, even more than here. So I encourage everyone to check it out. The web link is on the left and I'll present it again and again. Hopefully I can convince you all to jump on, take a look and give us some feedback. So the data in ICE is really diverse and it's organized by toxicity endpoint related to regulatory endpoints. So on the left side, you can see the organization of our various data. On the right, you can see examples of some of the assays that are listed within each of these toxicity endpoint categories. Today, I'll be focusing on the orange, which is the curated high throughput screening data. This is a curated version of the ToxCast and Tox21 assays, which are produced by an interagency collaboration. The Tox21, of course, includes NIH, FDA, EPA. The ToxCast comes out of EPA, and so we lump it all together and use EPA's process data to curate and provide an ICE as well. But there's a lot more there, so a lot of in vivo data and others, including predicted phys-chem properties and predicted toxicity endpoints like Karmaus, SERAP, and Compara that I would encourage everyone to take a look at. So talking about the high throughput screening data, the data are organized in a way that we can search in 2 different ways. This assay selection is been developed this way because different people have different questions and we want to be able to address user needs depending on how you want to access these data. The first is mechanistic target. So this is a biological process that the assay is informing on. And then there's mode of action, which is really taking that biological process and linking it more towards a mode that can contribute to an actual toxicity endpoint that we've organized the data into. So on the left side, there's what you can see under the search tool. When we select assays, just high throughput screening. I'm not interested in any other data. I don't even necessarily want to look for regulatory endpoint. I just want to search the in vitro high throughput screening data. This is ToxCast and Tox21. You'd be looking at the search box on the far left where we have mechanistic target-based searching. The blue tab shows you we've selected just the CHTS, and under it you can see these mechanistic target terms. Some of them are nested within each other. So cellular process has a little arrow because it has sub-terms that are more detailed. But this breaks you down into mechanistic, really focusing on assay target type of biology to identify assays you want to search for. On the right side, you can see an example of the mode of action-based search. In this case, I've clicked on the acute lethality tab of our select assays search window to say, I'm really focused on acute lethality, the LD50 endpoint. I want to know what in vitro assays might be contributing to it. Under acute lethality, we have a mode of action term, which I've expanded in the little window there, and it will show you the various mode of action biological grouping terms that have in some way been found by experts or literature to contribute towards acute lethality. This is really broad, it's not mechanistic, it's mode of action, because we want to be broad and inclusive on any assays that may be able to inform on contribution via these biological modes of action. So what I've clicked on in my example here at the bottom is I've chosen cytotoxicity as the example. And when you click on the information box next to cytotoxicity, this final little box on the far right shows up, and it shows cytotoxicity as a mode of action that describes assays contributing to anything related to cell survival, cell viability. And there are 117 assays. And these 117 assays have been mapped to 4 terms that all contribute to cytotoxicity. And on the right side are the links that take you to an ID for the NLM metathesaurus, where we've taken these exact terminology from. That gives you further definition of what constitutes cell survival, or cell viability process, or cell morphology, or what's cellular process, which is our catch-all for anything else under cytotoxicity. So imagine we choose our cytotoxicity box here. There's 4 terms within it. We know that all of those assays may contribute to acute lethality, and we conduct a search. This helps us because the mode of action groupings are supporting potential toxicological relevance. Of course, these are regulatory endpoints in particular that we're grouping these assays into, and it can help build a weight of evidence. As you group these assays together, all these 117 or sub-selection, if you choose to narrow that down, can help you with integrating various assays that before you might not have known are informing on this endpoint, or you might not have been able to interpret 117 assays worth of what is the target. This way you don't have to look one by one, it's giving you the groupings. The other thing that is helping here with regards to even the mechanistic target searching is understanding the biological process. So it's one thing that it can contribute to a toxicological endpoint of regulatory importance. It's another to say just general biology, I'm interested in anything estrogen receptor or anything endocrine, depending on how big or how small you want to make your search. You can produce a search on your specific chemical and say, wow, I was looking for estrogen and androgen, only estrogen results are coming. Now I've identified a data gap. There was no estrogen data for my chemical. The search has not only helped to pull data, but to identify what did not come as a result of your search criteria. So in that way, we're also helping identify data gaps and bring together assays that are informing complementary information. So in following through its example, here's an example result. You first get a summary data results landing page. In this case, you can see the bar graphs on the bottom. I've relabeled them so it's bigger, but you'd have the 4 different mode of action terms that came out with your search criteria. So in my search box, I put cytotoxicity. There are 4 different modes of action within the cytotoxicity term that contribute to acute lethality. I had a handful of chemicals that I put in. And here you can see that for my chemicals anyway, all the cytotoxicity information seems to be coming from cell survival and cell viability assays, which are the 2 bars on the far right that have a lot of higher number of endpoints, versus on the far left, the cellular process and cellular morphology assays don't seem to have a whole lot of data for the chemicals that I searched for. Now, when we look into these bars, we can see the biggest area is blue, and blue is inactive. So my chemicals were tested, but they were inactive. The red is going to be active, meaning there was an effect of my chemical in those in vitro assays. And then on the top, we've got some brown color, brown, gold, yellow, that's up to your interpretation, colors. The darker color is a QC omit. This comes from the curation that was conducted just for the ICE tool. The QC omit is going to tell you that there was a chemical QC concern with regards to some of the chemical samples tested. That information is downloadable, so you can see what the concern was. The TOX 21 program has conducted GCMS, mass spec, and other analyses to confirm the purity, the concentration of chemical being tested in the in vitro assay. And in this way, we're able to show you, well, it wasn't exactly necessarily pure or at the concentration that they thought. There was a warning about using the data. And the lighter color is the flag omit, which again is specific to ICE. We have developed a workflow based on expert knowledge of these assays and of assay performance, as well as the curve fitting algorithm, to identify when we think that a concentration response effect may not be robust and should be of concern. And I'll show you how to view what those are and why they might be of concern so that you can review it yourself to see if you agree or decide to use the data nonetheless. So in looking at all these bars, we get a really quick glimpse of where we have data, where we don't have data, how much activity or inactivity we may have, or how much concern there might be with your chemical purity or quality of the data. We can then look further by clicking on the actives. So this would be the red portion of the bar. And there's two ways we can look at it. On the far left, we've provided a bar graph where every single colored box plot is a different chemical. And every dot shows you where the AC50 is in a different assay. So you can see the spread of AC50, so potencies in different assays for your chemical. Which assay was the most sensitive? Which assay was the least sensitive? By putting my cursor over each of these dots, if I was live on the web, it would tell me what the assay name is. So you could see which of the assays are more sensitive or less sensitive for your chemical. On the right side, we can view it by assay instead. So by clicking the blue button on the bottom of the left, we would create the plot on the right. And the plot on the right, each box plot is actually an assay. So now you can say in this in vitro system, which chemical was the most potent or the least potent, and you can rank the chemicals from your search per assay. So this is giving users the ability to dive into it either from a chemical perspective or from an in vitro assay perspective and see how consistent or inconsistent assays or chemicals are within a more narrow field. Of course, we're pulling potentially up to 117 assays. So this way we can go one by one if there's interest to do so. Another thing we can do is look at a detailed view for a chemical. So this visualization tool was created because some people would like to see for one chemical of interest as much data as possible. So in this case, I've pulled BPA. Of course, we know that's very data rich. On the far left, you can see phys-chem properties. Some of them are predicted, some of them are experimental. Everything is these little green information circles. You put your cursor over it and it gives you information as to where that data comes from. But in this way, you can pull that data. Again, we have a summary to look at. I did a subset of assays just so we don't have too much data to look at, which mechanistic targets, biological groupings of assays have activity or no activity or lots of data or not lots of data. And that's what the bar graphs would reflect. And you can customize which assays or biologies you'd like to look at for your search. So this is just the substance level details. On the very top, there's two blue tabs. We're on the substance level, that's in blue. We can also jump over to the curve surfer. The curve surfer, you can see on the top now that tab is selected, is going to show you every single concentration response plot for that chemical in each of the assays tested. It defaults to only showing you active curves, but this is where you could choose, I want to see everything that ICE decided to flag with their curation. Why is it flagged? What do those curves look like? Do I agree that they should be flagged or not in my work? Which of the assays had really nice, robust sigmoidal curves? Which ones seem to have just a little bit of a blip at the top concentration? You can browse through and see every single one. The other nice thing about this tool is that in addition to the assay name, which sometimes is cryptic and hard to interpret, we've provided the mechanistic target for the assay so that you can quickly look at it and say, oh, this one came because it's related to cell viability process. So we're trying to help facilitate understanding a little bit more of the context because we understand that some of the assay names are not approachable to everyone. Not everybody's going to learn thousands of assays names and what that assay is informing on. We've provided context, and here we're providing a view of the actual data itself. There are dotted lines plotted across the horizontal axis. There's a dotted line showing where the top of curve is. The vertical dotted line is where the AC50 was drawn and derived from. And you can download all these images in high quality. You can also download spreadsheets with the outcomes of the search in its entirety. So this is my summary slide. I really just wanted to articulate that high throughput data from in vitro assays is high throughput. There's a lot of it. It can be really challenging and daunting to approach. But with this ICE web resource, we've integrated that data in a way that we've curated it to help gain confidence. We're very transparent about what flags are there, and you can review the data, you can review the flags in terms of what we suggest having caution for, whether it's chemical purity based or based on the response of the curve of the data. There's expert generated annotation of all the assays. You can look at it from a mechanistic, very focused mechanism detailed biological perspective, or from regulatory endpoint mode of action groupings in searching, so that you can build weight of evidence approaches or identify data gaps. The search can be done on either one of these groupings. And of course, we've developed multiple visualizations. I've shown you some screenshots today that show we've got summary across chemicals. We can do assay or chemical specific summaries. And of course, view the data itself, depending on how far in you want to go. You can look at the summary plots, or you can look at actual curves for any of the assay data at all. So one more plug, one more showing of that link on the top left here is that ICE website. There's so much more though. There's lots of in vivo data, as I showed earlier. There's much more data than just the high throughput screening. And there's also lots of tools. There's an IVIVE tool where you can put in that in vitro data that you searched for and identified based on the tools I showed today. And without having to generate a new search, you can click a button that sends it over to another tool. You've identified which of those curves you liked, which chemicals, which assays, throw it into the IVIVE tool, and that will do an in vitro to in vivo extrapolation and give you what the potential in vivo dose is of that AC50 that is derived from the in vitro assay. We also have a chemical characterization tool that talks about chemical use. Anyway, there's so much more. I really hope everyone will go play with it. And on my very last slide, this is our most recent publication that talks about some of the background on how we did this annotation, how we derived some of these tools. Feel free to read up on more detail. That's me if you want to email me. And of course, the ICE website and its email address. Suggestions for new tools, edits to the tools that are there, any challenges you have, we're happy to help and hear back from everyone. And with that, I'm going to say thank you for the time and for listening.
Well, thank you, Agnes. That was a great presentation. I have used the tool myself for a number of different applications. It's really useful. And so thanks for giving us a tour. We have a few questions, so I'll start to address them. Looking back, this one looks back to the slide where you were looking at the acute tox example, and they're asking, are the term definitions like cell viability coordinated with the existing NIH assay guidance manual?
So we're trying very hard to stay consistent within NTP and certainly NIEHS on how we apply our annotation vocabularies. They are controlled vocabulary terms from the NLM Metathesaurus. All of those can be linked out. We do have the links for it. And we're coordinating with SEBS to make sure that the way that we structure data can interact with theirs. We're trying as hard as we can to be coordinated. So that's the best I can say, I suppose, on that front. Yeah.
Another question is or is it possible maybe, or how can you annotate assays that could contribute to multiple modes of action, like cell viability and cellular process?
So assays are not restricted to being mapped to only one term. You will notice that if you search for one term and another term, the same assay is coming. And that's because we do recognize that they can contribute to more than one, and they are annotated to multiple. That does arise. So that's a great question, and we have tried our best to integrate that.
Okay, I've got a number of great questions. I'm just gonna check our time here. Okay, so Another person is asking for the potency box plots by assay or by chemical. Is it possible to change the y-axis to logarithmic?
So the y-axis will not change to logarithmic, but all plots generated, including the box plots, including the bar plots, every plot, are interactive. So you can click and zoom if you want to, and you can rearrange what section you're looking at. If it looks really clustered, you can zoom in on and see the points. The other thing you can do is download what is in the plot in a Excel spreadsheet tabularly and plot it yourself if you would like to manipulate just that subset of data. So it's very dynamic in terms of what you can look at or what you can download.
Great. For the AC50 assay data, is the cell type used listed?
Yes, we do have a download file that provides the cell type used for all of the assays. There's a lot more annotation available than what we've integrated into the dynamics of the search tool. I will say that in some cases in vitro assays, there's a cell system and then it's transfected with a receptor from a different species potentially. So I wouldn't get too hung up on the cell type, but that information is available. We do acknowledge people are interested in it, and it's available for download and is something that we have in the queue to add into the search so that you can filter and choose that as one of your criteria.
Interesting. Great.
Great question.
One last quick question I want to get in here. Can concentration dose curve analysis be done on ICE with my own data?
At the moment, we do not have the opportunity for uploading your own data on concentration response curves. You can upload your own data for running the IVE tool. Other tools have that functionality, but for viewing the curves and looking at our curation, that is very much based on a database that we have in the back end where we've provided the values and is not something you can apply live. But that's a great question and we'll certainly think about that for future tools. Thank you.
Great. Okay, well, I'm sorry we didn't get to all the questions. Maybe Agnes can type some answers.
And we can do batch searching. So I just see that now. But yes, upload your chemical lists. Okay, I'm done. Thank you.
Awesome. Thanks. Thanks very much. Okay, I'm going to turn it over now to our next presenter, Saskia Galanuk. If you would like to share your screen, you can take the floor.
Okay. Let me just turn on my laser pointer. You can see it, right?
No, we don't see your screen yet.
Yeah, you see?
No.
No. Okay. Oh no. I'm sorry.
There we go.
Ah, there we go. Sorry.
No worries.
Then I have to turn on the laser. Okay, now it works. Okay, first of all, thank you, Christy, for the kind introduction. And I also would like to thank Esther for awarding me with the Best Poster Award. And yeah, for giving me the opportunity to present my work today. The human-induced pluripotent stem cell test as an alternative method for developmental toxicity testing. So far, reproductive and developmental toxicity testing is mainly performed in vivo, according to OECD or US EPA guidelines. And these consume about 90% of all animals and 70% of costs used in regulatory toxicology. And due to these high animal numbers, accompanied by high cost and a low throughput, there's a strong need for alternative methods that allow for faster and more cost-effective assessment of toxicity. And one alternative test method for developmental toxicity testing is the mouse embryonic stem cell test. And this ECVAM validated test makes use of mouse embryonic stem cells formed into embryoid bodies, EBs, and then they are plated onto a matrix and spontaneously differentiate into contracting cardiomyocytes. And an endpoint analysis in the validated study are a cell viability assay and the assessment of beating structures after 10 days. And the heart is the first functional organ in the developing embryo. After the fusion of two endocardial tubes, it already starts beating after only 3 weeks of gestation, being in the developmental stage of a simple heart tube. And any disturbance in this early developmental process will lead to spontaneous abortion. However, species differences might hamper the prediction for humans. For example, some cardiac genes are expressed differently in mice and humans. Here, NIEHS and the NIEHS specific cardiomyocyte markers often used for molecular analysis. The predominant form in human embryo is NIEHS, with a later switch to NIEHS. But in mice, it's the exact opposite, with NIEHS being the predominant form in the embryo and the later switch to NIEHS. And furthermore, the response to substances is different as well. So for example, in this study, 150 compounds associated with significant human toxicity were tested in rodents and non-rodents, and the true positive results obtained from only rodents was about 40%. Data obtained from non-rodents led to a true positive result around 60%, and only both data combined resulted in a true positive result of 70%. Which would mean an even higher use of animals to obtain reliable data. And these results underline the need for alternative human-based in vitro methods. So to overcome these species differences, the aim of this study was to set up the HIPS test as a human cell-based alternative method for developmental toxicity testing to reduce and replace animal testing. And the basis of the HIPS test is a high-quality IPS culture. So we used cell line IPS11, and a high quality IPS culture is shown on the top row. The cells are small in size, have multiple nucleoli, and have a low cytoplasm to nucleus ratio. And on the bottom row is an example of differentiated IPS cells, which are not usable for downstream applications. Cells start to flatten out and build so-called differentiation cracks, which are seen as white lines in the intracellular space. And they have a high cytoplasm to nucleus ratio, also being enlarged. And the difference is especially seen in B and D, representing the same magnification here on the top right. And to further characterize the IPS culture we used, we performed flow cytometry analysis with different stem cell markers. So from A to D, you can see examples of IPS11 cells in passage 4, analyzed for the markers SSEA4, NANOC, OCT4, and SOX2. And all of them are above 90%. And in accordance with the literature, we avoid prolonged cultivation of IPS cells and decided to culture them for 10 passages. Because as you can see in E, marker expression slightly decreases over time. And notably, in passage 20, the threshold of 95% OCT4 positive cells is not reached anymore, which is proposed to be a threshold for a successful cardiomyocyte differentiation in the literature. And we perform cardiomyocyte differentiation with cells in every other passage, from P4 to P10 and additionally in P20. And while we obtained beating cardiomyocytes from IPS cells in passages 4 to 10, the cardiomyocyte differentiation was visibly impaired using IPS cells in a high passage like passage 20. And the quality of IPS11 cells we used was further controlled during the process of the banking as a master and working cell bank. So to induce the cardiac differentiation, the modulation of two pathways is important, namely the Wnt and the BMP pathway. So here you can see the Wnt pathway with no ligand present, and this is the degradation complex which continuously degrades beta-catenin, which cannot enter the nucleus. And in our protocol, we use a small molecule called CHIR to force activation of the Wnt pathway. Because CHIR blocks GSK3 as part of the degradation complex, and with this, beta-catenin can accumulate in the cell and enter the nucleus to activate target genes. And on this side, Wnt is present, which leads to the recruitment of the degradation complex. And to force an inhibition of the pathway, we use the small molecule IWP2. So IWP2 blocks porcupine, which normally palmitoylates the Wnt molecule. And without that, it cannot bind to its receptor. So in general, we have an activation on the left side with CHIR and an inhibition of the Wnt pathway using IWP2. And the BMP pathway is simply activated by adding BMP4, leading to the dimerization of SMAD1 and SMAD4 entering the nucleus. So being able to modulate these pathways, we adapted the protocol for cardiomyocyte differentiation from Zhang et al. So the protocol for the HIPS test takes 10 days in total, modulating the Wnt and BMP signaling pathways in a 2D culture. So on day 0, the cardiac induction is started by the addition of BMP4 and CHIR. And as Wnt has a biphasic role during heart development, the pathway is inhibited using IWP2 from day 2 to 4. And on day 7, first contracting areas are visible, synchronizing into a beating wave until day 10. And on day 10, we perform endpoint analysis for cell viability and video recordings for analyzing the beating frequency and the area, and collect samples for PCR analysis. Taking the variability of IPS cell lines into account, the protocol is adaptable to new cell lines, applying a concentration grid of BMP4 and CHIR on day 0 to get the highest amount of beating cardiomyocytes. So we did this for the IPS11 cell line, and the grid was performed on a 24-well plate with concentrations of CHIR ranging from 1 to 1.7 micromolar and BMP4 from 0.75 nanogram per milliliter to 2 nanogram per milliliter. And the initial optimal concentration combination for IPS11 cells was just on the edge of the grid. So therefore, we had to extend the grid with these concentrations here to ensure that the best concentration were chosen. And here you can see exemplary video recordings of the extended grid of day 10 cardiomyocytes. And as you can see, the concentration combinations marked in green leads to the most confluent cell layer of beating cardiomyocytes, which are 1.75 micromolar CHIR and 0.75 nanogram per milliliter BMP4. So after achieving the best concentrations of CHIR and BMP4, the differentiated cardiomyocytes were then characterized. So the morphologic development of cardiomyocytes was tracked using a microscope. And here you can see pictures from the same well from day 1 to day 10. So on day 1, cell-cell contact inhibition was not present anymore, allowing the cells to grow on top of each other. And until day 4, the cell growth continued irregularly. And you see these darker patches, which means that cells have grown on top of each other, have grown more on top of each other. And 2 days later, cells already start to form these holes, which are more present on day 7. And on day 7, cells already start to beat, detaching from the matrix. So from that point onwards, the morphology did not change that much until day 10. So to monitor the transition from pluripotent stem cells to cardiomyocytes, we performed PCR analysis over the time course of 10 days with different markers across cardiomyocyte developmental stages. So while stem cell marker OCT4 decreases, the earliest marker for cardiac mesoderm, MES1, shows a peak expression shortly after the activation of the Wnt and BMP pathways. And subsequently, ISL1, a cardiac progenitor marker, arises too. And both MES1 and ISL1 are downregulated during maturation, which is accompanied by an increased expression of the cardiac progenitor marker GATA4, which is expressed until day 10. And TNNT2 and alpha-actinin 2 specific cardiomyocyte markers are the latest to arise. And while TNNT2 is significantly upregulated from day 5 onwards, the same holds true for alpha-actinin beginning 2 days later from day 7 onwards. And the quantification of CTNT and alpha-actinin positive cells via flow cytometry shows that we have about 80% cardiomyocytes exemplary shown in A and B for day 10 cardiomyocytes. And to monitor the expression of these proteins during the development, flow cytometry was performed on days 8, 9, and 10. And although there is a tendency of increased expression of CTNT and alpha-actinin 2, it's not significant. So in addition, the majority of cardiomyocytes was evaluated using ICC. So staining for CTNT after 10 days of differentiation shows that, consistent with the flow cytometry results, almost every nucleus is attributable to a CTNT positive staining, which is better seen in the merge picture. And the staining also shows that we gain immature cardiomyocytes as no binucleation is seen and cells display disorganized sarcomeres. And for a better comparison, this is how organized sarcomeres look with the same marker in mouse iPS cells. And you can clearly see the striated structure here, which is not present in our cardiomyocytes yet. So taken together, the gene and protein expression data indicate a successful and stable cardiomyocyte differentiation for the HIPS test. And although gene and protein data indicate a stable expression of the evaluated markers from day 8 onwards, day 10 was chosen to evaluate the endpoints, as the beating is best recordable and most prominent on day 10. As you can see in days 8 and 9, the beating is almost unnoticeable and not synchronous yet, so you have multiple starting points. And on day 10, the cells start to beat in a noticeable synchronous wave, which can be analyzed with our in-house developed software. With that, next we challenged the protocol with compounds. So according to the mouse EST, we tested penicillin G as a negative control and 5-FU as a positive control. And penicillin G has no effect on cell viability, beating frequency, or beating area. And 5-FU is drug used for the treatment of cancer and is a known embryotoxic substance inhibiting proliferation. So 5-FU significantly reduces cell viability at 1 micromolar, which is accompanied by a reduction of the beating frequency and beating area as well. And compared to the mouse EST, the HIPS test is almost 3 times less sensitive in the endpoint cell viability. And furthermore, we established a PCR marker or PCR markers as a molecular readout. So looking at TN92 and alpha-actinin 2, we found that penicillin G has no effect on the expression of these genes. And 5-FU does not have an effect on TN92 either, but significantly decreases the expression of the later marker alpha-actinin 2 at 1 micromolar. So here I will shortly show you the advantages and disadvantages of the HIPS test. So the system is human cell-based, having no ethical concerns using human iPS cells. And we do not use FBS in any of our media, as the use is highly discussed due to its variability and the origin. Our protocol leads to a defined amount of around 70% cardiomyocytes on day 10. And due to its stability in cardiomyocyte generation, only 4 replicates per condition are needed. And furthermore, the evaluation of contracting cardiomyocytes is not human-based, using an in-house developed software. Disadvantages are, however, the use of a 2D system. And although only 4 replicates are needed, the assay is still considered as low throughput. And we use self-produced FTDA medium for the iPS culture, consisting of 10 different components, which increases the lot-to-lot variability, making the test very error-prone. But however, we are working on this point at the moment. So in summary, the HIPS test was established using a defined protocol, and molecular characterization indicates development via cardiac progenitor cells to immature cardiomyocytes. And endpoints for toxicity readouts include cell viability, cardiomyocyte-specific marker expression, beating frequency, and beating area. And so far, 5-FU and penicillin G were tested as positive and negative controls, respectively. And as I mentioned before, we are currently establishing a commercially available medium for the iPS culture to avoid the lot-to-lot variability in the FTDA medium. And yeah, so with this, I would like to thank my coworkers and the sponsors for their support, and you for your attention.
Great, thanks, Saskia. That was a great presentation. Let me ask everyone to put their questions in the Q&A box. We have about 10 minutes for Q&A. There's one question there already. It is, did you distinguish atrial versus ventricular cardiomyocytes?
Actually, no. I know there are protocols out there using, for example, activin A to force them into the atrial differentiation, but we don't know which cardiomyocytes we have to speak so we only know that they are beating.
Okay. And a follow-up question, how much maturity has been reached?
Well, assuming that our cardiomyocytes are in the developmental stage of this early heart tube, they are pretty much immature, I would say. Also, you could see it in the ICC staining. It's just kind of green. You can't see any structures within the sarcomeres. Yeah, so they are really disorganized, maybe. So they are for sure embryonic cardiomyocytes in a really early stage.
Mm-hmm. Another question is, how are you measuring the beating frequency and beating area?
Yes. So as I mentioned, it is an in-house developed software. I'm not sure if we have the time to explain this in detail. You can write me. I'm just noticed that I don't wrote an email down. So you can find me by Googling my name, I'm pretty sure. So it's basically, we analyze every pixel in the video and they change their color gradient. So we first transform them into a grayscale video, and every grayscale has a certain number, to say so. So there are 256 different grayscales. And as soon as it changes, it's noticed as a beating. So when you look at a Z plot, that would look like this, kind of like, yeah, so you have different peaks and you can distinguish the beating from the cells, actually, from dirt swimming around in the well, because when you have dirt in your well, which is recognized, it would be a plateau, which is not recognized as a peak. So a sharp peak is a beating in the Z plot. This is everything I can explain in this short amount of time, but if you're interested, we are also working on the software to improve it. So we actually found another way to analyze the beating area, which looks more promising so far. So this is all in preparation in a paper. So yeah.
Okay. Stay tuned.
It's kind of complicated. I'm not an expert in informatics. So.
Well, just to mention, Helena kindly put your email in the chat.
Oh, thank you very much.
If anyone wants to get in touch with Saskia to discuss more, you can. So the next question asks, did you benchmark your cells compared to commercially available cardiomyocytes? So we don't have commercially available cardiomyocytes because most of them are actually too old. And also our focus is, well, the development from iPS cells to cardiomyocytes. So it doesn't give us any advantage in buying cardiomyocytes, because we want to see how substances have an effect on the development from iPS cells to this early state of the heart tube. So it's embryotoxicity, not cardiotoxicity. This is really important, and sometimes people overlook this. Although I really talk a lot about cardiomyocytes, it's more like embryotoxicity, not cardiotoxicity, actually. So yeah, okay. We don't use BITE or commercially available cardiomyocytes. That makes sense. And maybe jumping off of that answer, the next question is, have you compared cardiac differentiation and neural differentiation to obtain better predictability?
Sorry again, cardiac and? Cardiac
differentiation and neural differentiation.
Oh, that's a good question. So our working group is actually working with different cells and cell lines. So the working group of Alan Fritsche is also heavily focusing on the neurosphere assay and working with neuronal cells. Also on the establishment from RPS cells to neuroprogenitor cells, differentiating them into neurons, astrocytes, oligodendrocytes. But this is in progress, I would say. But I have a colleague that is working on this. So basically our whole founders are split in two. So I'm the part from iPS cells to cardiomyocytes, and my colleague is working on the part from iPS cells to neurons, to say so.
Got it.
Yeah.
Great. I had a question about fetal bovine serum, and I'm wondering, that's a big issue in vitro toxicology, and I'm wondering if you just started from the very beginning avoiding bovine serum, or if you had to kind of transition your protocol or modify a protocol.
Actually, no. So we never ever used FBS from the beginning. So some protocols, I think I'm pretty lucky that I only use these early cardiomyocytes. I know a lot of protocols have to replate the cardiomyocytes and then use FBS. Actually, this protocol is also using FBS for later cardiomyocytes. I never tested culturing them for a longer period than 10 days. Yeah, so I'm not sure if it would work without FBS, but I know a lot of protocols using FBS for mature cardiomyocytes or getting them more mature, but I'm lucky enough.
This last question, I don't think you mentioned it in your presentation, but we'll see if you have any thoughts to share. And they're wondering how well the mouse embryonic stem cell tests predict the human positive compounds.
That's a good question, actually. So first of all, the mouse EST is also under development from other working groups. So the really kind of old validated test is only using a cell viability assay and also the evaluation of the contracting EBs after 10 days, which is also human-based. And there were a lot of improvements for this assay, actually. So I think with the prediction would be better using the additions of the mouse EST, I think. So we only tested penicillin G and 5FU so far, but well, penicillin G that did nothing in the mouse EST and in our assay. And regarding the cell viability, our assay is actually 3 times less sensitive than a mouse EST. What this means, we have to test it, further substances to see if our assay is in general less sensitive or just the specific substance. We don't know yet.
Okay. Well, we've gotten to the end of our questions just in time. So one thing I want to point out again is the chat, which Helena has put in that the journal ATLA, Alternatives to Laboratory Animals, is preparing a special issue on FBS-free approaches in case anyone's interested in that. So with that, I will thank all of you who presented, Agnes and Saskia and Helena, thanks for joining us. I want to thank all of you for listening in and taking the time to ask questions and be engaged in these presentations. It really makes for a great webinar when you get so many questions and have some discussion at the end of each presentation. And that's it for 2021 for ASCCT ESTIV webinar series. We'll see you back in January and hope everyone has a happy holiday and a wonderful new year. Thanks everyone, take care.